Hey everyone, just wanted to welcome you. We still have a couple people coming in, but we're going to get started in just a second. Okay, I think we've reached a critical mass, so I'm gonna go ahead and get started. So hi everyone, my name is Alex Sakel. I am the segment manager for oncology and cancer research here at PacBio, and I'm going to be your host for the webinar today. So I want to welcome all of you to our liquid biopsy webinar, and I'm really excited that you've decided to join us. So today I'm joined by my colleague Young Kim, who is the senior staff product manager and our resident Onso system expert. So we're both going to be presenting a presentation today, and those presentations will then be followed by a question and answer session. So you're welcome to submit your questions during the talks by typing them into the area provided on the attendee control panel, and then we'll get to the questions during the Q&A session. And so just so you know, we are recording this webinar, and we'll be making it available to you in the next week or so. So please be on the lookout for a follow-up email that will contain a link to the recording. In addition to that, we have also uploaded a number of complementary pieces of literature to the control panel, so please feel free to download those and take them with you. So with that, we're going to go ahead and get started. I will be presenting the first talk on the value of sequencing accuracy for liquid biopsy research. So I'm going to turn off my camera here to make sure the focus is on the slides, and then Young and I will both come back on video for the Q&A. Okay, so thanks so much everyone again for being here. I'm excited to tell you about PacBio's newest short read sequencing platform called Onso, and how our highly accurate sequencing by binding technology is improving rare variant detection in liquid biopsy research. So first, let's talk about accuracy. Most of you may be thinking of PacBio as a long read company, but we think of ourselves as an accuracy company. So we have the most accurate long read sequencer on the market, and we now also have the most accurate short read sequencer with Onso. So this is important since not all DNA that you might want to interrogate is long. So case in point is circulating tumor DNA, which we'll talk about today. And so when we think about short read sequencing technologies over time, there have been a huge reduction in the cost per gigabase, that's shown here in pink on the plot, with corresponding increases in the number of samples per year that can be run, shown in aqua. However, if you look at run accuracy, there has been very little improvement over time, which is particularly problematic for needle in a haystack type applications like ctDNA detection, where the noise due to sequencing errors can obscure the signal that you're trying to detect. So PacBio's newest sequencer, Onso, is positioned to fundamentally change that paradigm. So how much more accurate are we talking here? So I'm showing in this plot the empirical Q-scores per base. So Onso is in pink, Elements of VD is in purple, and Illumina's NovaSeq 6000 is in orange. And for all three technologies, these plots are from whole genome sequencing of the HG002 reference sample. So to orient you, on the x-axis is the cycle or base number, and on the y-axis is the mean empirical Q-score per base across all reads. And on the right, what I'm showing is what those Q-scores mean in terms of the probability of error. So Q30 is 1 in 1,000, Q40 1 in 10,000, Q50 1 in 100,000, and so on. So as you can see here, Onso's Q-scores are above Q50 for the majority of the read, and that comes out to an average error probability of 1 in 270,000. And you can also see that it doesn't show the characteristic drop in quality towards the ends of reads, which is the result of accumulated errors over time. And this is in contrast to the other technologies, which both have significantly lower Q-scores. Something to note here, you may notice that the first 30 bases are lower and then it becomes higher, but we've actually recently demonstrated that we are over-penalizing the Q-scores for those first 30 bases. And so we're in the process of recalibrating our Q-score algorithm, and we expect that the next iteration will show Q-scores above Q50 for essentially the entire read. So this is a huge improvement in accuracy over what is available today. So how do we accomplish this? It's in the chemistry, which we are terming sequencing by binding, as opposed to sequencing by synthesis, which is what most existing short read sequencing methods use. And so here on the left, what I'm showing is how sequencing by synthesis, the traditional method, works. First, you flow in a labeled nucleotide, which is bound and imaged. Then the fluorophore is cleaved, and the nucleotide itself is incorporated, which results in what you can see here as molecular scarring from the leftover linker arm that was attached to the fluorophore. And what this results in is the polymerase runs into these molecular scars and has a hard time dealing with them. And so that is what increases the error rates. By contrast, SBB decouples the interrogation and incorporation steps. So first you flow in that labeled nucleotide, it binds transiently and is imaged, then it's washed away, the blocker is removed, and an unlabeled nucleotide is flowed in to be bound. And then this results in the synthesis of a more natural DNA molecule. And additionally, we are able to separately optimize the chemistry for the interrogation and incorporation steps, as opposed to sequencing by synthesis, where they have to resort to some pretty harsh chemistries to do this cleavage and incorporation altogether. And so the combination of these two factors is what leads to the dramatic improvement in accuracy that we see with Onso. So what does this actually look like? Here on the right, I'm showing an IGV zoom in of a region with SBS on the top and SBB on the bottom. So if you aren't used to staring at IGVs all day long, to orient you, each gray bar here is a read, and each colored point indicates a base that is different from the reference sequence. And so you can see really clearly there's a ton of colored bases in SBS, where those are errors that are arising during sequencing, whereas for SBB, it is essentially error-free. And on the left here, I'm showing some of the benefits of SBB in addition to the accuracy in general, in terms of sequencing through repetitive regions, negligible index hopping and duplication rates, and lower input requirements. And Young is going to go through all of those in detail in his talk. For now, I'm going to focus on accuracy and how this impacts what we're able to do in liquid biopsy. So why do we care about improved sequencing accuracy for liquid biopsy? Here, what I'm showing is the typical patient journey with ctDNA levels in time, shown in green. And so you can see various time points of tumor growth, then you have surgery and ctDNA goes away, then perhaps you have recurrence followed by treatment. And so with SBS, the limit of detection due to sequencing error, many of the time points that you would sample during liquid biopsy would be undetectable. So for each of these tubes with an X through them, those are sampling time points at which the ctDNA levels are below the threshold that SBS is able to detect. In contrast, with SBB, with a 10 to 100-fold increase in accuracy, lowers that limit of detection. And what that means is that more of these time points are therefore detectable, which could potentially result in earlier detection of cancer or recurrence. Okay, so that all sounds great. Let's see what the data looks like. How does SBB actually perform for a needle in a haystack application like liquid biopsy? So to test this, we performed a head-to-head comparison of SBB versus SBS using a commercially available reference sample. This is the SeraSeq ctDNA complete mutation mix. So this reference sample contains 19 small variants at known variant allele frequencies. So there's 12 SNV, single nucleotide variants, 5 deletions, and 2 insertions. And these have been diluted in human wild-type DNA to mimic what you would get from a plasma extraction of ctDNA. And importantly, each variant allele frequency has been orthogonally validated by digital droplet PCR. So that's what I'm showing here on the table on the right. For each variant, you see the estimated allele frequency by digital PCR. And this is critically important because in order to determine how well we are doing, we need to have a really clear idea of what is present in the sample for us to detect. So we started with the reference material at different allele frequencies. Here we're looking at 0%, which is the wild type, 0.05%, 0.1%, and 0.25% allele frequency. And then to do this head-to-head comparison and eliminate potential variation due to sampling, we made use of our Onso library conversion kit, which Young will tell you more about later. So the workflow here is you take the SeraCare input different allele frequencies. We do library prep with the Agilent SureSelect XT HS2 kit, followed by hybridization capture with Agilent's Comprehensive Cancer Panel, which is a targeted panel that includes the variants present in these reference samples. Oh, sorry. And then the post-capture pool is split into two aliquots. So one of them goes directly into SBS, and the other is first converted via a simple PCR reaction to be Onso compatible, and then that goes into the SBB sequencing. And then so for each sequence sample following the sequencing, we did 10-fold subsampling to different sequencing depths of interest. This was to get a sense of the range of our estimates. And then we did variant detection with and without UMI, unique molecular identifier based error correction, and then looked at the sensitivity or recall of the known variants for each technology. Okay, so first we're going to take a look at a single variant site. So here I'm showing the IGV for the NRAS Q61R variant at 0.1% frequency. This is looking at the SBS data. And so if you look in the zoomed in box here, you can see that there are multiple sequencing errors surrounding this variant site. And then if you look in the table, you can see that we're actually getting base calls for all three alternative alleles here. Making it completely unclear which of these would be the true variant, or indeed if there is a variant here at all. Perhaps all of these are due to sequencing error. And you would have no way of knowing which of these is correct if we weren't looking at a reference sample for which we already knew what was in there. In contrast, if you look at the same site in the SBB data, you can see in this zoomed in box here that the IGV is completely clean, so there's no noise due to sequencing errors. And then if you look at the base counts in the table here, indeed, we are only getting calls for a single alternative allele, making it really clear that the true variant here is a T to C. So that was for a single variant. What does this look like across all variants? Here I'm showing a plot of the sensitivity or recall for three different allele frequencies. So this is the 0% wild type sample, the 0.05% sample, and the 0.1% sample. And recall, just to remind you, is the number of variants that we called correctly out of the total number that were there in the sample. So this is the percentage of the known variants in the reference sample that were called by each technology for each allele frequency. And then SBB here is in pink or magenta, and SBS is in orange. So what we see, these are results using UMI-based error correction for both technologies, is that SBB results in significantly higher recall at low variant allele frequencies compared to Illumina, even with half the sequencing depth. So you can see a significant improvement at these low frequency alleles in terms of recall. So then if we actually go down even further, here we're looking at 6,000x SBB compared to 24,000x SBS, even with fourfold less sequencing and not using any error correction. So SBB here is without error correction, where SBS is with error correction. Even with both of those things, SBB is still achieving similar or better recall rates compared to SBS. So then to dig into what is causing this increase in recall, here I'm showing a plot that compares the estimated or observed allele frequency for all variants, those that are called in the wild type, so any variant called in the wild type here is a false positive, versus those that are called in the sample. So in this case, this is the 0.1% allele frequency sample. And so for SBB, we see that there are only 2 false positives called in the wild type across all 10 subsampling runs. And something that's important to note here is that for clinical tests, typically your goal is to reduce false positives. So in our sample, we can only confidently call the variants that are above the allele frequency of the false positives in the wild type. So this is essentially your variant detection threshold that sets the allele frequency above which you are able to confidently call variants. So for SBB, none of our true variants are below this allele frequency cutoff, meaning that all of these would be able to be called. In contrast, for SBS here shown in orange, we have a ton of false positives in the wild type. And if we look at the estimated allele frequencies of those false positives, that significantly overlaps with the allele frequencies that are estimated in the sample. And so what that means is that none of these variants that are below this variant detection threshold for SBS would actually be able to be called. And this is significant because let's take this variant here, for example, it's very close to that threshold, but but it wouldn't be called given this threshold that we have to apply due to the false positives. And it could be that this variant is one that indicates that the tumor is developing resistance, or perhaps conversely, it's a variant that confers drug susceptibility. But either way, you would be missing this variant completely using existing technologies because of the high false positive rate in your wild type. So then just a final note here is that SBB also does a better job of estimating the true allele frequency of the variants. So here I'm showing a box plot version of the same data that was shown as points in the previous slide. So then SBB in pink here, the line in the center of the box plot is the mean of the distribution. And so you can see that for SBB, our observed or estimated allele frequency is essentially exactly at that of the samples. This is the 0.1% variant allele frequency sample, and our mean allele frequency is right at 0.1. In contrast with SBB, or SBS, excuse me, you can see that the mean allele frequency is higher than expected, and also that this distribution has a wider spread. And we believe that this is likely due to an overestimation of the true allele frequency in the SBS sample due to false positives that are being included in the estimation and inflating. So to summarize my portion of the presentation here, what we are seeing is that improved sequencing accuracy with SBB is changing the game for liquid biopsy by enabling reduced noise due to sequencing errors at variant sites, which leads to more confident variant calls. We see significantly better recall for low frequency variants, even with half the sequencing depth. And we see similar performance in variant detection, even with fourfold less sequencing and without UMI-based error correction. This increase in recall is likely due to the dramatic reduction in false positives in our wild type sample, which lowers the limit of detection for accurate variant calling. And finally, SBB's accuracy results in improved estimation of the true VAF, likely due to reducing inflation of the allele frequency estimate caused by sequencing errors. So thank you very much for your attention, and I will turn it over to Young for the second half.
Thanks, Alex, for providing a brief overview of SBB and how accurate sequencing can improve on detecting low frequency variants in liquid biopsy. So for this part of the webinar, I will walk through more details on the benefits of SBB and how the entire workflow can be easily implemented in today's NGS lab. So let's talk a little bit about the Onso system. So it is a mid-range sequencer with specifications similar to existing on-market systems. However, we differentiate versus other systems out there by the accuracy. So greater than or equal to 90% of bases are at Q40 or higher. That's one error out of 10,000 versus 1 in 1,000 with existing technologies. So this allows researchers to start tackling challenges limited by inherent sequencing errors that cannot be readily overcome by simply sequencing deeper. In addition, we are providing a solution that allows conversion of existing P5, P7 adapter containing libraries to SBB compatible version through a very simple and easy to use library conversion kit, which we anticipate will be a significant part of the initial testing to compare existing SBS versus SBB data. So as Alex showed this slide previously, we see numerous benefits of SBB over traditional on-market short-read sequencing platforms. Our combination of innovations in clustering and sequencing chemistry really delivers notable advantages over existing on-market sequencing platforms. So we'll spend some time here going over these benefits in the subsequent slides. So a benefit of SBB has been our ability to sequence through difficult, low complexity and repetitive regions such as homopolymers, which are often difficult to correctly enumerate using existing sequencing by synthesis chemistries due to it being a known error mode for the chemistry. Hence, many homopolymer sequences will need to be investigated further for their accuracy. So here, we show that across most homopolymer lengths on the x-axis, and the fraction of those lengths that are known to be correct on the y-axis, SBB in magenta clearly demonstrates better enumeration of the bases, while SBS in orange suffers from errors as a function of a homopolymer length. So how is this relevant to potential translational applications such as oncology? So there's a growing evidence for homopolymers and simple repeats such as microsatellites and its classification as microsatellite instability, whether it's high or it's stable, being a potential biomarker for selecting individuals for immunotherapy in oncology testing. And those repeat sequences, like microsatellites, are often difficult to read through with SBS due to slippage events. So we've seen with SVB, it can better resolve these microsatellites, hence providing a more accurate assessment of an individual's microsatellite status. In general, we believe homopolymer error correction has multiple utility, from polishing existing reference genomes to other research applications, such as inherited disease research, which we'll show an example next. So here's a clinically relevant example of SVB's ability to resolve homopolymers. The TOM40 locus shown here is a challenging region to sequence due to the presence of poly-T repeats, as you can see here, from anywhere between 19 base pairs to greater than 30 base pairs, depending on the individual. So it's been shown that longer repeats have been associated with late onset Alzheimer's disease, hence a potential biomarker to accurately quantify the repeat lengths. So SBS reads on the top here in the target region is unable to provide high quality reads, hence accurate quantification of poly-T repeat units, while SVB on the bottom is not only able to clearly enumerate, but also show heterozygosity in the T repeats. In this case, a T16 and a T35 genotype, which is indicative of a carrier for this poly-T polymorphism. We'll talk next about negligible index hopping as seen for SVB. So a major advantage of SVB versus SBS platform is the negligible rates of index hopping as seen, a phenomenon common with SBS platforms using pattern flow cells and exclusion amp chemistries, whereby presence of free adapters lead to increased index misassignment of libraries and or increased filtering out of reads, resulting in data loss. Due to SVB's approach of rolling circle amplification, RCA, for cluster generation, improperly paired indices are not exponentially propagated, hence a significant reduction in indices with incorrectly assigned indices. And our internal results indicate an estimated index hopping rate well below 0.01% compared to SBS. Hence, you can be confident indices are correctly paired with libraries, and DMUX results faithfully represent the intended index library pairing. Next, we'll move on to low duplication rates. So we also see decreased duplication rates versus SBS, especially optical duplicates and platform duplicates caused by idiosyncratic chemistry artifacts specific to newer generation of SBS sequencers. So shown on the figure on the left are all sources of potential duplication events, with optical category mainly impacting systems with non-patterned or random flow cells, as it color-coded here, while platform occurs with flow cells that have pattern flow cells in chemistry. And others here are shown in the blue occur in all types of platforms. The SBS cluster generation process, SVB, cluster generation process is done in a random order flow cell with RCA as described earlier. Compared to newer SBS instruments with pattern flow cell plus exclusion amp chemistry instruments, where molecules from one well can be released into solution and bind to another adjacent empty well. And these types of duplicate events are negligible in SVB. So besides PCR duplications where duplicates are necessary to amplify small amounts of input DNA, we see other types of duplicates here are really wasted sequencing since no additional information is obtained, hence increasing the overall cost of the read. And lastly, we'll focus a little bit on lower input requirements. So we've seen SVB chemistry and workflow has been able to drive down the input material needed for clustering and sequencing. So based on our internal testing of input amounts going into cluster generation versus different SBS platforms, as you can see here, we definitely see an advantage here on overall input needed by a factor of 7 to 10-fold. So the upside here is really using less library for increased flexibility for library optimization or titration experiments, and also ability to rerun the libraries as needed without having to redo the library prep, especially if those samples are really precious. In many cases, many clinical samples typically are. So let's switch gears and talk a little bit about how our solutions fits in with the existing NGS ecosystem. So our real focus is in the middle part here, really putting together the market-leading library prep, sequencing, and data output solution. And through our ecosystem partnerships, as you can see here, we have some of the top vendors in the library prep and secondary analysis space whose solutions are compatible with Onso. So it's of note that this ecosystem really has been in play for over the past decade. And as such, we see customers have capabilities really to take raw FASTQs to BAMs and finally variant call files to derive biologically meaningful data. As such, we don't need to reinvent the wheel here, and we have not seen pipelines break on Onso using commonly used tools that customers have. There may be room for optimization and tuning of the SBB data type because it's highly accurate and really understand error profiles. But overall, it is really out of the box compatible to ensure we get to results quickly with Onso. So what we're offering with Onso is a comprehensive solution from library prep, conversion kits, sequencing system, and associated consumables. And really backed by years of commercial experience and service and support and a global presence to boot. Through our experience on the long-read platforms and understanding of the sequencing technologies over the years to help better position long-read versus existing short-read platforms, I feel we're confident to deliver the same level of expertise and knowledge to the short-read side of the business. And enabling workflows and experience in line with what short-read customers expect is a key part of the Onso experience, with the value add being extraordinary levels of accuracy to enable more discoveries. So here's an outline of the Onso sequencing setup and run. The experience of really interacting with the sequencer and consumables should require minimal lift for all users alike. The Onso platform uses an on-screen guided setup to instruct users in a step-by-step manner from loading of the sequencing consumables and flow cell to starting the run. So we anticipate most users, experienced or novice, will find the process pretty intuitive. And our feedback from our beta testing for Onso have indeed confirmed this. And from a data output perspective, Onso generates base calls to industry standard FASTQ format in line with existing current on-market systems. Hence can be used as starting input for a wide range of different secondary analysis tools for alignment and variant calling currently available in the short read ecosystem. It is really, at the end of the day, all about simplicity and compatibility, and really designed to get you the highest quality sequencing data with minimal learning and effort. Finally, let's talk a little bit about library prep solution. And you might wonder why there needs to be another library prep kit when there are perfectly good solutions in the current market for short read sequencers that we all know. Well, believe it or not, I think it's important to look at accuracy from not just a sequencer perspective, but from the overall workflow aspect. And library prep is where many errors are introduced, including end repair processes for standard TA ligation to append adapters. So in order to take full advantage of an accurate sequencer, we've engineered the library prep kits to deliver on a much higher Q standard to fit the accuracy narrative of Onso. So with this improvement, now we're seeing raw read accuracies boosted from around Q40 to Q50 using our native library prep solutions that reduce the number of library prep errors and artifacts, which to our best knowledge have not been addressed with commercially available library prep systems. So with our index adapters, really delivering on negligible index hopping rates and a library conversion kit that enables easy comparison with existing data at still Q40 plus quality, we believe you have a choice. And the choice is flexibility on whichever applications you need to run. And it all done at extraordinary levels of accuracy. So in summary, from our section really see Onso's extraordinary levels of accuracy, enabling researchers to get more out of their data with less compromises than existing on-market platforms, and a workflow that should feel at home with most NGS users. And we anticipate Onso's accuracy to increase discoveries of existing needle in a haystack applications such as liquid biopsy, as Alex mentioned, and also potentially open doors for merging applications where high levels of accuracy are needed. And we are starting to witness a new accuracy era in the horizon with an interest in sequencing accuracy. And what we hope that innovative tools like Onso can contribute to significant discoveries to the genomics community that will help further our scientific knowledge base. And so with that, that's going to conclude today's presentation sections for today's webinar. And Alex and I really appreciate you taking the time to join us today. And so with that, we'll move on to the Q&A section for this presentation.
Okay. Awesome. Thank you so much, everybody, for attending. Young and I are going to come back on camera now and start going through some of the questions that came in during the Q&A. And so again, if you think of anything that you would like to ask, please feel free to drop it in the questions box, and we will make our way through them. So briefly, just to start, there's a couple of questions on the workflow that we used for the liquid biopsy assay. So one question is, what specifically was the kit used to do the capture? So this was the Agilent's ClearSeq comprehensive cancer panel. And so I think that targets like 150 genes, of which obviously we did not need to sequence all of them for our handful of variants, but it did include regions that covered the target regions where our variants were located. Okay, here's one for Young. It sounds like we mentioned that we have both a conversion kit and a native library prep kit. So what would you say is the advantage or the use case for each of those?
Yes, the conversion kit is really designed to enable direct comparison of existing P5, P7 libraries and convert that to Onso compatible libraries for sequencing. And so that it really enables a direct comparison of SBS data with the Onso data. And so that's the major use case for that. With respect to the native library kit, again, that's where you really want to look at the workflow. And as I discussed earlier, that it's really about trying to get the highest levels of accuracy possible. So again, with our library prep, being able to suppress many of the ligation issues with respect to end repair, we generally see that using a native solution in our case really tamps down on certain types of errors that can carry through and show up on your results. So that's, I think, if you're looking for the highest level of accuracy, we should definitely say that the native library is the one to use.
Awesome, thank you. Okay, here's another question on what input DNA did we have to use with the comprehensive cancer kit to achieve this low limit of detection? So in this assay, we used an input amount of 30 nanograms with the idea that this is a amount that is achievable given standard volumes of blood draws and typical extraction kits. But we're actually now in the process of doing sort of a much broader sort of matrix of different input amounts and different sequencing depths to see how that affects our overall limit of detection. Okay, here's one for Young. Does Onso have any commercial kits for specific targets already?
Yeah, so I guess the question here is referring to, do we have a specific panel for target genes? And so we don't currently have a commercial kit for that now. But what we believe is one of those things where we can leverage the existing ecosystem, right? So a lot of our partners are indeed panel makers, right? That's what they do best. And so I think for any sort of catalog panels or commercial panels, it'd be best to reach out to them to deliver on that solution. So I think we're not really focused on the panel because there's such a broad availability for those panels right now. So we're just really focusing on the sequencing and the library prep side of things.
Awesome. Okay, here is another question. think this one's probably for me. So which bioinformatics pipeline would you recommend using for low frequency variant calling plus UMIs? And which did you use to analyze the SEER-seq data set? So as Young mentioned, so the data output is the standard industry standard FASTQ file. And so theoretically, and we have seen this in our collaborations using multiple different pipelines, that by and large, your existing pipeline should be able to be applied to the Onso data directly. In this case, we used Sention for UMI deduplication, but we've also used other UMI deduplication tools to great effect. And then you can, things like vardict for known variant regions is a typical variant caller that we've used. But again, in the context of collaborations, we've seen a wide variety of different tools used, and really, on the whole, they're able to handle the Onso data with very minimal tweaks. So I think you should theoretically be able to use what you are using already. Okay. Another question, did you test the system on real clinical samples, and how does it perform on low quality DNA, for example, from formalin fixed samples? Yes, this is a thing that in the long read space, we have long wished that people would stop putting their samples in formalin and destroying them. But so yes, this is a thing that we have looked into. We've looked into, we have tested this on clinical samples, both liquid biopsy samples as well as FFPE. And one thing that Young alluded to is that when your sequencer is this accurate, the errors that you see are actually those that have arisen prior to the sequencing. So either during sample processing or library prep. And so for FFPE, that is where the majority of errors are introduced. And so with FFPE samples, there is sometimes the need for some of those error correction based tools to deal with the artifacts that are introduced upstream. But we are still seeing that we see an advantage in sequencing accuracy in terms of detecting things that are currently below the limit of detection. So for example, in an FFPE sample, if you had something like a resistance mutation that was at very low frequency that you wouldn't be able to detect confidently. So we are in the process of really sort of doing extensive testing on various different sample types and trying to get a real handle on what our advantage is in each of those sample types. So there should be more to come there very soon. Okay, let's see. Another question on the bioinformatics workflow and pipeline. What do labs need to have in place for bioinformatics? I think this is sort of going back to the same point about you should be able to use what you use already, unless this was more specifically about what kind of computing infrastructure do you need in order to run Onso, in which case, Young, could you speak to that?
Yeah, so basically a lot of the analysis is done through a external PC, analysis PC. So a lot of the base calling and even the base call to FASTQ generation can be done here. Or you have optionality of setting up an external server to really convert the base call files to FASTQ externally, right? So you can push that out to an external unit to do so. So we have all the information in our site prep guide that tells you what kind of compute resources you'll need to enable full analysis of the data sets.
Okay, here is another question. Since turnaround time is important for liquid biopsy, what is the run time? That's a good question. Obviously, for clinical samples, the time to answer is very important. Currently, with our chemistry, because we are separating that incorporation and interrogation and incorporation step, there is a slight increase in the overall runtime. So for a 2x150 paired end run, the runtime currently is 48 hours. But one thing that I will point out is you may or may not have seen recently, we announced that PacBio just completed an acquisition of a company called Apton, and their key technology is extremely high throughput and high resolution optics, as well as microscopy scanners. And so with that, we hope to make use of that technology to dramatically increase both the throughput and the speed of our SBP platforms. And so that is currently in the process of being commercialized. And so we expect that this turnaround time should improve dramatically as we bring that up to date. Okay. Let's see. This is probably for Young, since it's going a little bit beyond cancer. What are the current disease applications for Onso?
Yeah, I'm not sure exactly what do you mean by disease here, but we can talk a little bit about applications. So obviously, as we're doing this webinar, we're really focused on oncology and ctDNA detection and liquid biopsy sort of samples are obviously top of mind. But as a mid-range sequencer, we have to support sort of the ecosystem of current applications that are currently being done on these sequencers. So things like whole exome sequencing, targeted panels, whole genome sequencing, which is not really the target application, but people want to use that to kind of compare Onso versus some of the other existing platforms that are out there. And so I think those are the majority of sort of applications. We also done some single cell RNA-seq work in partnership with 10x Genomics to ensure compatibility of that. But we believe that Onso should be able to support all the different types of short read applications that are really common today. Whether it may not be done in a native way, but it could be done through conversion, for example, right? Where you can convert those libraries as long as they have a P5, P7 adapter sequence.
Okay, here is another question for Young, I believe. How does the system deal with low complexity libraries, and what's the minimum number of samples that you could sequence on one flow cell? So could it be a single library, and would it need then like a spike in control?
Yeah, so we're not immune to some of the issues that Illumina sequencing has with respect to low complexity libraries. We do have to spike in some additional control, which is our Lambda QC to increase the diversity. So I think from that perspective, you would have to increase the control spike in a bit to kind of deal with low complexity libraries. And with respect to minimum number of samples on a flow cell, you could. I mean, obviously it depends on what the depth of sequencing you need on an output. So for example, on if you're doing a whole genome sequencing run, HG002, and you want a 30x coverage, a single flow cell would be able to at most do one sample, assuming you're doing a paired end 2x150 run, which the rated spec for that would be around 120 to 150 gigabases. And so you just have to calculate how many samples you can run by looking at the specs for our flow cells, and then obviously dividing by whatever coverage you need for your particular sample and your panel size. So that's all variable, and we can help answer any questions regarding that if you have any.
Cool. Okay, so here's a sort of general question. Do you know of any labs or institutions that are currently using PacBio ctDNA sequencing in clinical settings? So in this case, I think our lawyers would like me to stress that we are currently research use only. However, we are working closely with multiple different institutions to evaluate the use of Onso for ctDNA sequencing on clinical samples. And then our ultimate goal would then be to ideally work towards, sort of IDT or IVT lab developed test type of settings before we would then move into the clinical realm. But for now, we are research use only, and we are investigating this in the context of research. Okay, here's a kind of interesting one. Which step of your liquid biopsy workflow is considered the most difficult part, and how did you counter it? That's a good question, and I guess it depends on what you mean by difficult. One thing that we have, and this kind of goes back to the question of native library prep versus the library conversion kit, is that when you're trying to do these head-to-head comparisons, if you were to take your sample and split it to go into the two different workflows at the very beginning, you'll introduce variability due to essentially pipetting. So like you pipette and you may grab one set of molecules to go in one tube and another set of molecules to go in the other tube. And so then if you're trying to do the head-to-head comparison, you're sort of confounded by what differences are due to the actual sequencing technology and what are just a result of random sampling noise. And so that was a thing that it presents a challenge specifically for doing these head-to-head comparisons. And so that's where we were glad to be able to take advantage of the conversion kit, because then that basically you've taken it further along before you then split, and so that reduces the variability. Yeah, that was probably the most the largest intellectual challenge when we were setting out to do these comparisons, was how are we going to make sure that we were doing an apples to apples comparison. In terms of the actual workflow itself, as far as extraction, library prep, capture, those are all sort of very straightforward and in line with existing methods. Yeah, so I think the biggest challenge was really sort of in the design of the experiment and figuring out how we were going to be sure that what we were comparing was the most relevant comparison. Oh, here's another one for Young. Can you run libraries with different read lengths together?
Yeah, that's a good question. So basically, our flow cell design or sequencer design is able to do individually addressable lanes. They're not independent flow cells. So in an independent flow cell setting, you could run libraries of different read lengths together. But in our current setup right now, it's individually addressable. So basically, that means you can multiplex different sets of libraries into the separate lanes. We have 2 lanes on our flow cell, but they have to be run at the same time and run at the same read length configuration. So we're also obviously looking into future things where independent loading could occur as well. But for now, it's individually addressable lanes.
Okay. Oh, one last question. Can I join your team as a researcher? Thank you so much. We are glad that you feel that way. So if you are referring to can you work with us as a collaborator, we are definitely interested and actively seeking collaborations for people who have interesting samples and interesting questions where they think that the accuracy of Onso would really make a difference. If you're talking about applying for a job, please check our job page, because we definitely do have positions that are open, and we would love to talk to you. But yeah, in terms of a collaboration, if you have samples or a project that you think is really interesting, we would love to hear from you, and to please reach out. I believe there is a link that is in the webinar registration page that has a click here to talk to a scientist. And so if you go there and fill out that form, then we will receive that and we'll be able to get back to you. And then alternatively, you can please feel free to reach out to me directly. My email address is just and then my last name at pacb.com. So please, yeah, if you have interesting questions, we'd love to hear from you.
Seems like there's another one here in green for me.
Oh, thank you. Yep.
So the question is, how does Onso handle the AAV ITRs, internal tandem repeats, or any high GC-rich regions? So the first part is that we haven't currently sequenced the ITRs yet. So it'll be kind of interesting how that would be able to be handled by the chemistry. But here is one of those things I'd like to put a plug in for our long read side. I think in multiple demonstrations of our Sequel2E and Revio platforms really being able to go through these regions and suss them out. So I think that we would recommend the long read platforms for this particular application. And in general, GC-rich regions, I think we do fairly well, and we're also currently in the process of just kind of addressing a little bit of the really, really high GC regions, like greater than 80% and whatnot, through updated chemistries. So I think that you're going to hear soon in the next few months or so that we'll have some updates that would really help improve on this aspect as well.
Okay, give it just another little bit to see if there's any more questions coming in. But if not, we can go ahead and wrap up and give you a few minutes back in your day. So thanks to all of you for the great questions. And also thank you to Young for sharing his expertise and his experience today. And thanks to all of you for joining us. So then in closing, I just wanted to briefly mention 3 things again. So in the next week or so, we will send out an email with a link to this webinar recording in case you want to go back and listen to any of it again. I'm sure you do. Second, you can keep up to date on additional PacBio webinars and events by visiting the pacb.com/events page. And then finally, immediately following this webinar, you'll receive a short questionnaire. Should take less than 2 minutes. And we would really appreciate that if you do take the 2 minutes to fill it out, as it helps to understand your needs and what you liked and didn't like about this, and then helps us to plan for future webinars. So with that, thank you so much again to everyone for joining us today, and I hope we will see you on a future webinar or in the context of a collaboration. So with that, please take care and have a wonderful day.